Hi, my name is Sudithi Manthati, and this is my research project on understanding the effects of curcumin on zebrafish eggs as a model of cancer. Cancer is the second leading cause of death worldwide, and this is because cancer cells mutate uncontrollably and spread to other parts of the body. Because it is difficult to always test treatments directly on cancer cells, researchers have identified zebrafish embryos as a good model organism of cancer. This is because zebrafish and humans share an 84% similarity in genome sequencing, and also most zebrafish genes have a human counterpart. Additionally, they found that it is extremely similar to the mutations present in cancer cells. In healthy cells in humans, the p53 gene regulates the cell cycle. When this is mutated, it forms the p53 proto-oncogene, which can no longer regulate the cell cycle, allowing for mutated cells to continue to divide and forming a cancerous tumor. Researchers have noted similarities in the overexpression of the p53 proto-oncogene in leukemia and the development of zebrafish embryos. When this proto-oncogene is suppressed, gross mutations of the embryo result, allowing for researchers to easily observe the effect of treatments on cell signaling pathways. Additionally, I also researched curcumin, which is the active ingredient in turmeric. As an anti-cancer agent, curcumin has the ability to induce apoptosis or cell death and inhibit or stop cancerous cells from proliferating or continuing to grow through the suppression of cell signaling pathways, which once again is regulated by the regular p53 gene but cannot be regulated when it is mutated and overexpressed as a proto-oncogene. This allowed to the formation of my question. What is the effect of varying concentrations of curcumin on zebrafish eggs that are used as a model of cancer to display the inhibition of the p53 proto-oncogene? Based on my research, I formed my hypothesis that if the concentration of curcumin extracted from turmeric is increased, then the extracted substance would have more notable effects on the inhibition of the p53 proto-oncogene present in developing zebrafish, as seen through embryonic mutations. After this, I identified my experimental groups. My control group would be embryos that received no curcumin extract and only distilled water. This is representative of a cancer cell being allowed to continue to divide because it has not received any treatment that suppresses the cell signaling pathways. My experimental groups were the groups of embryos that received concentrated curcumin extracted from different amounts of turmeric ranging from 1 gram, 2.5 grams, 5 grams, and 10 grams. My independent variable was the amount of extracted curcumin that the embryos were exposed to, and my dependent variable was the severity of the mutation in zebrafish embryos. The main parts of my experiment were as follows. First, I extracted curcumin from different amounts of turmeric, 1 gram, 2.5 grams, 5 grams, and 10 grams, using acetone. In order to ensure that the concentration of curcumin was increasing as the amount of turmeric I used increased, I graphed an absorbance slope in order to ensure that as the absorbance continues to decrease, then the amount of turmeric has also decreased, therefore proving that as the amount of turmeric increased, there is an increased concentration of curcumin. I also bred my zebrafish and collected the eggs. The viable eggs were pipetted into petri dishes with extracted curcumin and water from the fish tanks 24 hours post-fertilization. I then viewed the phenotypes after 48 and 72 hours post-fertilization to observe the severity of mutations. This is representative of a zebrafish embryo 24 hours post-fertilization. This is what all my embryos looked like when I started my experiment. This is a control group, which represents the embryos with normal development after 72 hours post-fertilization. This is representative of a p53 proto-oncogene being overexpressed and not being inhibited by any treatment, which means that in a cancer cell, it would be allowed to continue to divide. Here, there can be normal sac development, normal body development, and normal tail development, all of which prove that no cell signaling pathway was inhibited. In 1 gram of concentrated curcumin, there is a slight effect as seen by the curcumin. There is shorter body length, slight stack mutation, a slightly bent tail, and spinal column curving. However, none of these are quite severe, and most of the embryos look similar to the control group, proving that curcumin did have an effect, however not extremely strong, with 1 gram. In 2.5 grams and 5 grams, there can be an increased rate of mutation. However, it is not as extreme as what was observed in 10 grams of concentrated curcumin, where there is severe sac mutation, a greater amount of spinal column curving, a severe hook tail, and extremely obvious shorter body length. This proves that as the concentration of curcumin was increased, the mutations also increased, which means that the p53 proto-oncogene that is being overexpressed in zebrafish embryos was clearly suppressed, thus suppressing the cell signaling pathways, not allowing for normal body development. I also graphed my results in order to provide another view at how the increased amount of curcumin affected the rate of mutation. Here I graphed based on mild, moderate, and severe mutations. The severity of the mutation increasing represents an increased inhibition of the p53 proto-oncogene that is overexpressed, which also demonstrates that in cancer cells, if curcumin was exposed, that they would not be able to divide because the p53 proto-oncogene would be suppressed and the cell signaling pathway would be more regulated, not allowing for the mutated cell to continue to divide and proliferate. All the trials in my experiment represent some sign of mutation compared to the control group that only received distilled water. Some sources of error, however, were present in my experiment. Some of the pipetting may have not been exact, so the exact concentration of curcumin per dish may have varied slightly. This may have caused different mutations that decrease the amount of severity that was noted in the graph. Additionally, because all the embryos came from the same parents, there is very little genetic variation, which must be considered as cancer affects all with different genetic variations differently. Because everyone has different DNA that is being replicated, and when the p53 proto-oncogene cannot regulate it in one patient, there may be different mutations present in the cancer as compared to another patient with the same type of cancer. This leads to what I would like to take on in future research for my project. One thing I would like to do is consider sex differences into account by using full-grown fish. It has been noted in healthcare recently that treatments affect different patients of different sexes differently. This cannot be observed in embryos as sex differences cannot be easily noted. However, in full-grown fish, there is still an overexpression of the p53 proto-oncogene. And when the different concentrations of curcumin are exposed to these adult fish, it could work more efficiently on one sex than the other, and the mutations may occur differently, proving that there is a different concentration of curcumin needed or different treatments are needed for different sexes. Additionally, if I had the chance, I would like to observe the effect of my experiment on actual tumor cells, because curcumin may have different effects on the rate of cell division and metastasis on an actual tumor, as it could be more aggressive and mutate at a much more faster rate than what zebrafish eggs develop at. Additionally, cancer cells have other mutations present, which I could not have specifically targeted through the zebrafish embryos, which is why I would like to try to introduce curcumin to actual tumor cells to see what specifically it is inhibiting during the cell signaling pathways. These are some of my selected references, and I would like to thank you for listening to my project.